Our next speaker is Master Student Brendan O'Brien. While on a foray during the Mycological Society of America meeting in Alberta, Canada, Brendan became so immersed in a conversation about applied mycological research with mushroom guru, Paul Stamets. They both completely forgot to look for mushrooms, lost their group, missed the bus, and Paul made an international call to reassure his wife. Hey, Dusty, it's me. I seem to have gotten lost in the forest of Alberta looking for mushrooms. But don't worry, it's okay. I'm with Brendan. While on an exploratory expedition to survey rainforest fungi in the Medidi region of the Bolivian Amazon, Brendan was forced to abandon a collection bestowed to him by a village shaman after being repeatedly threatened by two competing bands of howler and capuchin monkeys. Brendan ran away and the dominant species took the specimen. While crossing the Arctic refuge by foot and packcraft, Brendan discovered flawless porcini specimen grown on a ridgeline in association with dwarf birch. When he came across a group of native Alaskans hunting caribou and moose, he suggested they trade sources of protein. The villagers let Brendan keep his mushrooms, but fed him the best caribou steak nothing can buy. Please welcome Brendan.
Thanks. So I'll reveal which one of those stories is untrue when the presentation comes to a conclusion. So yeah, I'm Brendan O'Brien. Thank you all for coming. I am a student in the Nutrient Cycling and Ecological Design Lab with Dr. Eric Roy. The title of my work is Physiochemical Properties of Combined Dairy Manure and Food Waste Digestates and Their Use for Cultivation of Edible Mushrooms. I'd like to just first thank the Rubenstein School and Casella Resource Solutions for supporting my work. So I first came to Rubenstein School because I was looking for an opportunity that I was unable to find elsewhere. I became inspired by the role that fungi play as primary decomposers in our environment and was interested in harnessing this potential in an applied way to integrate them into human recycling systems of scale. As you can see, fungi are useful. For many centuries, fungi have been utilized as food and medicine, and more recently, their mycelium has been employed to create biodegradable alternatives to building insulation and packaging materials. Fungi produce extracellular enzymes that break down the complex biopolymers that constitute plant structural proteins, and these are, of course, lignin and cellulose. These compounds are quite similar in their chemistry to persistent organic pollutants in the environment, and as a result, these enzymes have really wonderful implications in terms of their use in ecological restoration. These same enzymes have also been used to treat different forms of plant biomass for digester feedstocks to enhance the availability of carbon to the microbes in the digester. So I perceive the field of applied mycology as something that is infinitely useful, and that's why I'm here at Rubenstein to explore a way that we can integrate fungi into human recycling systems. So the primary question that my research focuses on is food waste and using this food waste to in turn grow food. So can we turn this highly heterogeneous, putrid, gross stuff that comes off the back of a Hannaford dumpster truck back into a useful, nutritious food product in the form of fungi? So as we think about a circular nutrient economy, the question I'm asking is, how can we maximize the value of nutrients contained in food waste to, again, grow food within our existing food system? So many of you are probably aware of Vermont's Act 148 Universal Recycling and Composting Law. This law is a series of measures that is instituted over several years, starting when it was first passed in 2012, and finally culminates in a full food waste ban from landfills in 2020. To help guide management of food waste, the state published the food recovery hierarchy. And you'll notice that below source reduction and food for people is diverting food to animals and compost, and then anaerobic digestion, and also energy recovery. But all of those, well, I'll just say those three options, diverting food to animals, to feed animals, and composting and anaerobic digestion, result in byproducts that are ultimately land applied. So important take-homes are that greater than 50% of Vermont's total waste stream can be diverted from landfills, and about a third of the total waste stream is organic material that can be recycled for energy production or nutrient recovery. Similar laws have also been passed in Massachusetts, Connecticut, and Rhode Island, indicating a regional trend towards increasing organics recycling. As I mentioned, anaerobic digestion is one such method of recycling these nutrients for biogas production. This is a desirable method because the infrastructure is already in place. We have 17 operational dairy farm manure digesters on farms around Vermont, and there are over 21 in the New England area. Again, recycling food waste in this way is a method of capturing greater amounts of energy from the material, but it also results in byproducts that are ultimately destined for land application. This particular digester is a facility in Maine that accepts food waste from over 150 different Hannaford supermarkets around the Northeast. But again, while this boosts energy production, these byproducts that are likely a result, or that are a result, need a use. And biogas production is not a nutrient management solution. It's an energy recovery solution. So in thinking about nutrients, this is a schematic that describes the flow of nutrients on the current dairy farm system. The primary inputs are feed and bedding material that go into the husbandry of the cows, who then produce manure, and soiled bedding, which becomes the primary feedstock for the anaerobic digester, which, as I mentioned, produces biogas for energy production and heat, which can be captured. And then the resulting byproducts can be separated, the resulting sludge can be separated into both solid and liquid fractions that have different agronomic potentials. The liquids are most typically applied to the landscape as fertilizer for feed crops and pasture fields. Some of that feed is exported off the farm. Most of it is recycled back to the cows. And so really the only nutrient output is lost to the environment, which results in eutrophication risk downstream. So if we integrate the current dairy farm system with our organics recycling system, and we introduce food processing residuals and food scraps to this system, that represents a new pool of nutrients that is entering the farm landscape that farmers are going to be responsible for managing. Again, this has the benefit of boosting biogas production for energy, but is also likely to increase the amount of amendment product as a result. I forgot to mention that the solids are typically recycled as bedding material on the farm, and ultimately then back into the digester. Much of those nutrients are leached back into the liquid fraction and ultimately land applied. Very little of these products are sold off the farm, and so we can expect that most of the nutrients in this system are winding up back on the farm landscape. So what are the risks of introducing this new pool of nutrients to the farm landscape? Are some of these materials more prone to leaking nutrients into the environment than others? As we know in Vermont, land application of excess nutrients in the past has resulted in eutrophication in Lake Champlain, and all of these organics recycling strategies that I've mentioned result in end products that are destined for land application. And the outputs of these are expected to increase as we introduce new material to the farm landscape. So what that means is we need a new strategy to produce food and also limit nutrient losses to the environment. So how will the addition of food waste affect the usefulness of these digestion byproducts? This visual comparison shows a clear contrast in the material from both dairy manure and food waste feedstocks, and one that is just from a dairy manure feedstock. So visually speaking, these are different materials, and their nutrient profiles are also expected to be different as well. Again, when you produce methane in an anaerobic process, your main product is methane, which is carbon and hydrogen, and so those nutrients are conserved in the residual byproducts. So the first phase of my research is to compile a database about the nutrient content of these digestate materials. So I'll be doing a literature review and providing a survey to operators to collect historical information that they have of their materials themselves. I have gone with the help of Adrian, also in the Nutrient Cycling and Ecological Design Lab, to sample 5 different farms, 2 of which are accepting less than 1% food waste into their feedstock, and 2 of which are accepting between 25 and 40% food waste. And then I'm visiting one farm that is in the process of scaling up their the amount of food waste that they're accepting as feedstock into their digester. So there'll be a before and after sampling as well. So the first phase is to reduce uncertainty around the nutrient variability of these materials by collecting information on what exactly they are. And I'm treating both solids and liquids, or not treating, but measuring the nutrients in both solids and liquids, and doing 3 different phosphorus extractions that will get at different forms of phosphorus of varying solubility to kind of get an assessment for the availability to plants once applied to the landscape as fertilizer and their potential risk to water quality downstream. I'm also looking at the organic matter content of the solids to get a sense for their usefulness as in terms of a compost ingredient, bulking agent, or digester feedstock. This is a preliminary comparison of 2 of the solids, one from just a straight dairy manure feedstock and one from a manure and food waste. And you can see that the manure and food waste digestate is much more enriched in both nitrogen and phosphorus. And especially in phosphorus, it's almost 3 times, as a percentage of dry matter, the amount contained in the just dairy-based manure solids. So what if we can divert this material before applying it to the landscape to produce food? And this is where the fungi come in. So my idea is to integrate fungi into the organics recycling system to produce food and capture some of this value of the recycled nutrients before they wind up in the landscape. So if we integrate within the organics recycling system more food waste, we have more amendment product, potentially more eutrophication risk, and then we integrate this mushroom farm plug-in, is what I propose, and we're left over with mushrooms, which is a high-value, nutritious protein source that can then be exported off of the farm landscape, fed to people. And then we have the residual material, which is called spent mushroom substrate, and I'll be characterizing that in terms of its nutrient quality and content, and also the organic matter quality as well, to determine if it's best recycled as a composting ingredient or a digester feedstock. So in the process of mushroom farming, this does require an energy input. Hopefully that can be offset by the boost in energy production from food waste. And there is hopefully some value in this residual spent mushroom substrate with regards to its use as a compost ingredient or digester feedstock, because again, the digestion of the substrate by the fungi has likely changed the organic fractions and made it more available to microbes in a composting or anaerobic digestion application. Phase 2 is to conduct a mushroom cultivation experiment. Well, I will experiment with 2 different digestates, one of a low food waste content and one with high food waste, compare those to a control, and test the amount, test the yield and biological efficiency of the mushrooms produced at 5 different concentrations to get a sense for the most efficient use that mushroom farmers might get out of this material. Also be analyzing the tissue of the fruiting bodies themselves to gain a sense of just how nutritious or the potential risks if they bioaccumulate metals, and then, as I mentioned before, assessing the spent mushroom substrate itself. I had the opportunity to pilot this project with the ecological design course taught by Eric Roy, my advisor, and we had a really good time in this class collecting these solids off of a farm, inoculating them at various concentrations in a sawdust media, and what we found that was as we increased the concentration of solids in the substrate, we got more mushrooms. So we increased the biological efficiency and the yield as we increased the concentration of digestate in the mix. So the overall goals of this work are to integrate fungi into the organics recycling system in Vermont, to minimize environmental harm by diverting nutrients away from the landscape, harvesting them as food, and then to test the residual spent mushroom substrate as a compost ingredient, bulking agent, or digester feedstock to determine its potential value and usefulness, and then also to produce safe, nutritious, high-value, protein-rich food. I'd like to just thank the Rubenstein School, Dr. Eric Roy, and the Nutrient Cycling Ecological Design Lab, Casella Resource Solutions, and all of my farmer collaborators. Any questions? None at all? Adrian.
Yeah, how do you expect the spent
I expect that the yields will correlate to the ratios of carbon to nitrogen to phosphorus in the material, and the more enriched, i.e. food waste containing digestates, will likely have a greater effect on mushroom yield at lower concentrations in the mixture, and those from just a dairy manure based feedstock will also increase yield, but at a lesser concentration, or at a higher concentration rather. I may?
I guess I was wondering about the comment you made where after you grow in the mushrooms you have a higher quality substrate left, because I would think after mushroom growth in something like sawdust that they would have been able to suck out all the high quality stuff, you harvest it, and you get left with leftover lignin type substrates that are hard to decompose by microbes.
Sure. And that is part of the question, is what is the value of the spent mushroom substrate with regards to digester feedstock or compost ingredient? A lot of composters out there are dealing with this food waste material as well, which is really high in nutrients and kind of low in carbon. So a material that is basically just stable carbon could be really useful to composters as well. Does that answer your question?
Sure.
Jed?
I'm going to ask a real quick one. Really quick. Are you experimenting with different types or species of mushroom here?
No, I'm working exclusively with Pleurotus austriatus, the common oyster mushroom, because that is essentially the go-to workhorse of all experimental substrates. It will pretty much eat anything. So, okay, thanks a lot.